All right, here's question 5 of the 2022 National 5 biology paper. And we've got a genetic engineering question. So this is one where we have to work out how substance is produced by genetic engineering and talk through the various parts of that process. So let's have a look at the question. Penicillin was the first antibiotic used to treat bacterial infections. Right. Bacteria have been genetically engineered to produce large quantities of penicillin. The diagram represents some stages of this process. Okay, now you probably haven't learned this in your biology course, but penicillin originates in a fungus or in a mold. And so this is going to be a fungal cell or a mold cell. And the gene is extracted, or the DNA is extracted, and then the gene is cut out of the DNA and inserted into this structure. This structure comes from a bacterial cell. It's a plasmid, it's taken out, and then the plasmid is cut and used for inserting the gene. Then the plasmid with the inserted gene, or the modified gene, or the modified plasmid or the engineered plasmid is inserted back into a new bacterial cell, another type of bacterial cell, which is cultured to produce the protein. Okay, so let's have a look at the questions. Using the information from the diagram, complete the table. All right, which one is the bacterial cell? Well, this is where you probably could run into trouble here. Now, you're going to get a mark for each of these, so if you got one wrong, it's not going to negate your marks. So the bacterial cell is this one. Now, you might think it was this one, because if you don't know out of which organism penicillin comes, you might think it comes from bacteria. But stick to what you know. You know that the plasmid has to come out of a bacterial cell. So pick the one that you know is a bacterial cell. So E has to be a bacterial cell. This is technically a fungal cell, I think, in terms of penicillin. All right, so that's E. The gene for penicillin is going to be this one then. This is C. It's labeled there. That's the gene, and we're going to cut that out and put it into the plasmid. And D is a plasmid, and you can probably say engineered or modified plasmid. We'll call it modified plasmid. But you will get the mark for just plasmid. Okay. And then it says, describe the next stage in the production of penicillin. Well, the modified plasmid, we just said this already, is inserted into a bacterial cell, not the same one, so don't say the bacterial cell, and don't say, so into a bacterial cell, and cultured to produce the protein. Okay, so where are you going to go wrong in this question? Well, there's that one there, but it will only cost you 1 mark because this table, quite often you see these tables are only getting 1 mark, but this has asked you for 3 pieces of information and giving you 3 marks. So there you go. There you are. Question 5. That's it. Nice and straightforward. You just needed to know your genetic engineering stuff. That's the tricky bit there. It's not clear to you. You won't have learned probably in your National 5 course that penicillin comes from fungi, and you might think it comes from bacteria, so you might choose that as a bacterial cell. Just stick with what you're sure about. The plasmid has to come from a bacterial cell. Pick E, not A. You don't need to pick A because you know that E is a bacterial cell. There you go. All done. See you next time.